Hey everyone, welcome back. So today's video is going to be about photosynthesis, and we'll also talk about the factors which affect the rate of photosynthesis. Because when we talk about the rate of something, we are obviously talking about the speed, which means how the things are happening per unit time. So topic is photosynthesis rate and factors affecting it. As a general overview, we know that a leaf gets water from roots and ultimately through the process of transpiration pull. It also needs ions, which include magnesium and nitrate, most specifically, which come here towards the leaf through xylem. And then finally, leaf has veins, which helps in the movement of water towards all the mesophyll cells. We have already talked about the structure of leaf, so we know that how individual cells get water. Another thing we know the leaf needs for photosynthesis includes carbon dioxide gas. Atmosphere has a carbon dioxide percentage of 0.04%. And this carbon dioxide enters the leaf through the stomata. We know the process. Light falls on the leaf, which is a very important requirement because the word photo itself means that it needs light. Oxygen is a byproduct of photosynthesis, so all the oxygen produced is eliminated by the process of diffusion. This is all happening during the daytime because during the daytime photosynthesis is happening in the first place. When we talk about the rate of photosynthesis, you can see a very clearly drawn diagram, which is a very good setup to measure the rate of photosynthesis. We have a beaker. On the inside, we have a water plant, for example, hydrilla. Then we have a filter funnel, which is obviously inverted upside down. Then we have a test tube, which is inverted upside down and placed on the top of the funnel. The water contains sodium hydrogen carbonate to provide the best concentration of carbon dioxide because sodium hydrogen carbonate produces carbon dioxide. I've already added this powder in water and mixed it well so that the water is rich in carbon dioxide concentration. When light falls on my apparatus, my plant will be able to photosynthesize. It will be absorbing carbon dioxide, light and water, but it will be producing a byproduct gas, which is the oxygen gas. So let me show you the bubbles of oxygen coming in. Oxygen gas bubbles are coming over here, which means that oxygen will obviously be lighter than water and it's going to be collected on top of the inverted test tube. So oxygen is collected on the inverted test tube. I can keep a track of time to see how much oxygen is collected per unit time. And as oxygen is collected, I can keep a track of the centimeter cube per unit time. For me, rate is equals to the bubbles of oxygen per unit time. If I do not have a very good apparatus to measure the volume, I can count the bubbles. Or more specifically, I can say the volume of oxygen gas in centimeter cube collected per unit time. Imagine three different pieces of apparatus. I'm going to talk about the role of light intensity here. So these three different apparatus have three different light bulbs. These lamps are placed at the same distance each time. The plant is exactly the same, but you can clearly see in the diagram, the one on the left and the one on the right, they have different light intensities because the power of the bulb is different. 40 watt bulb provides lesser light intensity. 80 watt bulb provides a higher light intensity and 120 watt bulb provides even higher light intensity. Imagine the amount of carbon dioxide and access to water is same. The only factor changing is light intensity, so I am making a graph and placing light intensity in arbitrary units on the x-axis. By the way, the SI unit for light intensity is candela, but your O-level biology syllabus never mentions it, so don't worry about it. On the y-axis, I have the rate of photosynthesis, let's say the oxygen's volume collected per unit hour. I got a graph which increases and then plateaus and then becomes flat. In the beginning, it's a slanted line, which means as light intensity increases on the x-axis, the rate also increases, which means light intensity was limiting factor. A limiting factor means when you provide more light intensity, more chlorophyll was excited, more chlorophyll was illuminated by the light, and more photosynthesis could happen even faster. But at a certain point, the graph becomes a flat line. It means light intensity is no more limiting factor. It means light intensity cannot control the rate anymore. Even if you increase the light intensity, the line is not increasing, the line becomes flat. It means increasing light intensity does not increase the rate even more. It means the ball is now out of the court of light. You can't control the rate by light anymore because all the chlorophyll have been illuminated, they all have been excited. So there has to be some other factor. What other factor could be there? We'll talk about it. The maximum rate, the maximum speed of photosynthesis has been achieved. Now, even if you increase more light intensity, it doesn't make a difference. We need to understand graphs like these. And to understand, let's say carbon dioxide was the limiting factor, or maybe temperature was. Anything could be the limiting factor after the line becomes a horizontal flat line. Because light intensity is not the limiting factor, so anything else can be. You can see the purple line, it shows us that. Let's study another graph where again I have the light intensity in arbitrary units on the x-axis and I have the rate of photosynthesis as oxygen collected per unit hour on the y-axis. Faster photosynthesis will give me higher oxygen collection and vice versa. You can see I have 4 different lines. Each line is increasing then becoming a flat horizontal line. On the x-axis, as the light intensity increases, the rate increases in every case. There is no doubt about that. Orange, green, purple, blue, all lines are increasing in the beginning. Let's label the first line O as orange, the P is purple, we'll call it B and G for blue and green. When we compare orange and purple, orange has to have low carbon dioxide levels compared to P, because that is why P has a higher overall photosynthetic rate. In graph O, carbon dioxide levels were the limiting factor. And you can see when the carbon dioxide was increased for graph P, the purple graph showed a better photosynthetic rate. Let's compare the blue graph, which is B. You can see the blue graph has performed even better. That could also be because of carbon dioxide level. Maybe carbon dioxide levels were better for the graph with blue line. If we trace temperature and if we say that temperature was a limiting factor, you can see O and P had same temperature. B had a better temperature, so B performed better, and green, which is the G graph, it had the best temperature, so it performed the best. The point is that carbon dioxide and temperature also affect the rate of photosynthesis. In O, P and B, temperature was also a limiting factor. After a certain point. So temperature is also a limiting factor. If you provide better temperature, you get better results. Higher temperature provides more faster enzymatic reaction because all these reactions are catalyzed by enzymes. So higher temperature is even better. But make sure that higher temperature has a limit of 37 degree. It's better not to go beyond that. Another very important thing to notice is that when the line is slanted, when the line is steep, this is the slanted portion of the line. During this region of the graph, x-axis is your limiting factor, which is light intensity. Remember, during the slanted portion of the line, when it is a little having this angle, x-axis is the limiting factor. When the line becomes horizontal, it means anything could be the limiting factor, but the x-axis can't be the limiting factor. So during the horizontal part of the graph, anything but the x-axis is limiting factor, and during the slanted portion, x-axis is exactly the limiting factor. This will definitely help you a lot in your graphical analysis questions. Stay tuned, guys. Thanks.